In this lecture, we're going to talk about taste and smell. So let's talk about smell first. Well, smell is really an airborne molecule that comes into contact with your olfactory receptors. And so a molecule is actually a chemical. And so it triggers the olfactory receptors, and they're going to send a message in the form of a receptor potential to the olfactory bulb, which is cranial nerve 1. And then cranial nerve 1 is going to send a message to the brain. And so the olfactory receptors are chemoreceptors, and they respond to airborne chemicals. You have over 500 different types of olfactory receptors. And it's pretty amazing if you compare that with, say, taste. You have only 5 taste receptors. Okay, so it is well known that you can't taste things as well if you have a stuffy nose, or if you plug your nose, hold your nose, you can't really taste things as well. And so that's because your sense of smell helps you with taste. And so it is obviously you don't have 5 simply different tastes that you experience. So those over 500 different olfactory receptors really help with the different tastes of food that you experience. And likewise, the smell, your sense of smell, triggers memories. So, I still think of my father's second wife every time I pass by somebody wearing a particular perfume that she always wore. And so that's because the olfactory bulb sends nerve impulses to a part of the brain known as the limbic system. And the limbic system is sort of a centralized brain area composed of a lot of different structures, such as the cingulate gyrus, the fornix, the hippocampus, the amygdala, hypothalamus, and lots of different structures involved in the limbic system, and they are all involved in emotions. So it's the olfactory organs that contain the olfactory receptors. And let's look at the anatomy of smell. So the olfactory receptors are located in the superior portion of the nasal cavity. Okay, and so they are actually embedded in an epithelial lining that and the neurons hang down through the ethmoid bone, which is perforated. And then just above the ethmoid bone is the olfactory bulb, which is going to be sending messages to the brain. And remember that olfactory bulb is cranial nerve number 1 because it's the most anterior cranial nerve. And so what is going to happen is a molecule is going to bind to a membrane receptor, which is located on a receptor cell. And the receptor cell is actually a bipolar neuron that has little cilia that are hanging down into the superior part of the nasal cavity. And these cilia, being surrounded by epithelial cells, form a mucous membrane. And so molecules that enter your nose as gases then dissolve in the watery mucus fluid that is produced by the mucous membrane. And then they are going to bind to these receptor cells that are going to trigger a receptor potential. But one question many people have is, how do these receptor cells actually allow us to smell so many different things, because even though we know you have over 500 receptor cells, there are probably more than 500 different smells that you have experienced in your lifetime. So how do they actually work? One way is that a particular odorant molecule might bind to one receptor with a very high affinity and perhaps bind to another receptor with a very low affinity. And so it is thought that it's possible that a particular pattern of activity of different receptors is going to be what is sent to the or through the olfactory bulb. And so you have this single molecule that is binding to a number of different receptors in slightly different ways that end up giving a sort of characteristic fingerprint for a particular odor. The olfactory bulb is going to be sending impulses to an area of the brain called the prefrontal cortex. And the interesting thing about this is that the prefrontal cortex also receives information regarding taste. And it's thought that that may be one reason why olfactory stimulation while you're eating, so when you're smelling the food, that those smells can be perceived as taste as well as smell. And then taste sensations are going to operate through taste buds. So taste buds are the special organs of taste, and they are actually clusters of epithelial cells that function as a receptor or as receptors. And so they actually look a little bit like layers of an onion, which we'll talk about. So your tongue, which is shown here, has these bumps on it called papillae. And so if we take one papillus and we blow it up, and let's just look at it. Okay, there it is. And now in yellow, I'm drawing these little taste buds on the sides of the papillus. And then if we blow up one of the taste buds, you can see that it sort of looks like an onion. Layers of an onion, and what I am drawing are these taste cells, sometimes called gustatory cells. And the gustatory cells are the cells that function as receptors. So it's estimated that we have roughly 10,000 taste buds that, and that each taste bud has 50 to 150 taste cells. Okay, and then let's look at the remainder of the structure here. And so we see that there are these support cells for the taste bud. And then you have these taste hairs that are sort of microvilli that are attached to each of the taste cells that protrude from the ends of the taste cells out through this sort of taste pore into your mouth, where they can function as receptors to pick up molecules that have dissolved in your saliva. And then the binding of a molecule to the receptor is then going to trigger a receptor potential, and that receptor potential will then be carried on neurons to the brain. So you have 5 taste sensations, and those are sweet, salty, bitter, sour, and umami. And umami is sort of a savory or meaty flavor, which is triggered by amino acids, which, as you know, make up proteins. And realize that all taste buds can receive or respond to all the different taste sensations. And so this is quite a bit different from what we used to think, which was that different parts of the tongue responded to different types of taste. But now we know that each taste bud can respond to more than one taste sensation. But each taste cell is only going to be sensitive to one type of sensation. So it's either a sweet cell or a salty cell, but not both. And then each taste cell is going to activate a sensory neuron that is going to send that particular action potential regarding that particular taste, or specific taste, to the brain. So we have some examples on this slide of taste. So carbohydrates are going to activate the sweet receptors, and saccharin, which is a sugar substitute, can activate the sweet receptors. Very high concentrations of saccharin will give you a bitter taste, which is why people get sort of a bitter aftertaste if they are drinking diet sodas, because they have too much saccharin activating, which will activate their bitter receptors. And so realize that different concentrations of these different food chemicals will activate different receptors, based on their concentration. And then next you have the hydrogen ions or acids, which will give you a sour taste. Sodium and potassium salts will give you salty taste, activate those receptors. Magnesium and calcium also activate bitter. And it's been found that these different tastes are going to depolarize the cell in some way or by some mechanism. If it's sodium and potassium, it's about opening ion channels. If it is about hydrogen ion or acid, it is going to depolarize based on ion channels. The others are going to trigger receptors that are associated with G proteins. And then the activation of G proteins is going to lead to many different types of second messenger systems. And then also we forgot to mention that amino acids are going to trigger the umami receptor, which is also associated with G proteins. And that's the end of our lecture on taste and smell.